Welcome. This is the Chemistry, Biology, and Math Revision Hub. Today we are discussing the Pearson Edexcel International GCSE 9-1 Biology Paper 2B for January 2022. This is the part 2 of this paper, so I discussed already part 1, and I will put the link to that video in the description box. So let us begin with the first question. Question 4 says, fish farming is often used to produce protein-rich food. Selective breeding is often used to produce fish that grow rapidly and do not waste much food. Explain how selective breeding can be used to produce fish that grow rapidly. So here we need to start off by selecting fish that grow rapidly, and then we will mate those specific fish until we can produce offsprings, and then we will mate their offsprings to produce offsprings of those so basically we're going to repeat this process for several generations in order to ensure that the only alleles we are left with are those with fast growing fish. So I say by selecting fish that grow quickly, mate them to produce offsprings, and then those offsprings created are going to be mated, and then their offsprings, so basically they're going to be mated and they'll grow quickly, and then repeat the mating process for several generations so that alleles for fast growing or fast growth are passed on, and then we can guarantee that we have the fast growing fish. The next part says fish farming systems can often release ammonia into the water. The ammonia is converted into nitrates. Describe how ammonia is converted into nitrates. Now you need to know ammonia can be converted to nitrites by nitrifying bacteria, which later convert nitrites into nitrates. So I say nitrifying bacteria convert ammonia into nitrites, and then the nitrite into nitrate. That is basically it. You remember that from the nitrogen cycle. The next part says, multi-trophic level aquaculture is a method of fish farming that has been developed to reduce environmental pollution and increase profits. The diagram shows the multi-trophic level aquaculture system. So here we can see there are fish growing here, and we see the fish waste or waste food as well as feces. When they come out, they fall down into a different cage, and also the minerals can be leached to the seaweed, which is growing on floating platforms. In here, we can see the cage with lobsters and crabs that consume the waste food from the feces, or waste food as well as feces. So basically, this is a self-sustaining system whereby the waste coming from the fish is eaten by lobsters, while the nitrates that are passed on, or minerals, are going to be sent off to the seaweed so that they can be able to grow. So the next page says, explain how the multi-trophic level aquaculture system reduces environmental pollution and increases the profits of fish farming. So here we have to use information from the diagram and your own knowledge to support your answer. This is a 5-mark question, so we have to go all the way. And again, before we answer it, I want to take you back to the diagram so that we can be able to look and see clearly. So here we can see the fish here, the waste food as well as feces eaten by the crabs, and then here the minerals leaching away to the seaweed that is growing on the other side. So I said feces and waste food from the fish are removed by the lobsters. So here we'll have less decomposition by decomposers, and therefore fewer bacteria will grow in the water. Remember, if more bacteria grow in the water where the fish are, it might cause some problems. Next, I said less respiration by decomposers. So this is competition for oxygen. If there is less aerobic respiration by decomposers in the area where the fish are growing, it means less oxygen is going to be removed by the decomposers, so there will be less infection due to bacteria. The next part, I say the minerals like nitrates are removed by seaweed, so there will be less algal growth. Remember, nitrates are gonna stimulate growth of algae. So if they are removed, there will be less algal growth, there will be less eutrophication, and then we can have more products for the farmer, because some of these seaweeds could be later processed and used as food. So there will be no need to buy food for the crabs as well as lobsters. So remember, crabs and lobsters can also be sold. So if they are consuming the waste from the fish as well as the feces, then it's going to be a self-sustaining system. So this brings us to the end of question 4. Let's go to question 5. Question 5. The diagram shows a nephron from a kidney with 3 different areas. There is area X, Y, and Z. So X here, this is the entrance to the first coiled tubule, and then here the entrance to the descending loop, and here we have the collecting duct. So here they said the table gives the concentration of glucose and urea at X, Y, and Z. So here, the X area, we have 90% glucose. You can see the concentration is really high because reabsorption has not that much occurred. And then we have the concentration of urea on this side, which is kind of not as much as you go to point Y and Z. Now you look at point Y, 0 glucose concentration, and that's 0 at Z as well. So this is telling you at this point X, reabsorption of glucose is just beginning, or is just basically in the process of beginning. However, at point Y, all the glucose has been reabsorbed, so we have 0. Yet at this point, urea, the concentration of urea is just lower, and it's going to be the highest here due to excess reabsorption of water. So they say explain the difference in concentration of glucose between X and Y. And again, to take you back, this is point X and that is point Y. This is going to be due to selective reabsorption. So I say there is more glucose at point X than at point Y. So remember they said explain the difference. So you have to state the difference and then give a reason why the difference is the way it is. So my statement is this. I say there is more glucose at X than at Y because glucose is selectively reabsorbed in the proximal convoluted tubule, or you can just say fast coil tubule, by active transport using energy from ATP. The key words are the ones I underline. Selective reabsorption by active transport using energy from ATP. That is your explanation. So let's continue to the next part. Here they say, calculate how many times more concentrated the urea is in area Z compared to area Y. And they want you to give your answers to two significant figures. So here I began by looking at the concentrations per 100 centimeters cubed. At Y it was 440 and at Z it was 1700. So here I did 1700 divided by 440, giving me 3.86, which I rounded off to 3.9 since they told me my answer should be into significant figures. So here I say so Z is 3.9 times more concentrated with urea than Y. The key thing here was to find the concentration at Z divided by the concentration of at Y so that you can know how many times more concentrated Z is than at Y. So it's 3.9 times more concentrated. The next part says, explain the difference in urea concentration in the filtration and in the filtrate found in Y and Z. Now here I say, as you saw previously, Z has a higher concentration of urea than Y. That is the statement. Then the reason here we can see because water reabsorption. You remember at the end in the collecting duct there, by the time the solution or the liquid reaches or urine reaches the collecting duct, there's been more water reabsorption. So we still have the same number of moles of urea. However, there is more extracted water, meaning the concentration of urea will go higher since the volume has become smaller. So here I said, because water is reabsorbed in the collecting ducts by osmosis, because ADH hormone, and again this is an extra information, antidiuretic hormone is present to stimulate the collecting ducts to absorb more water, producing more concentrated urine. I hope you can remember that to cause the collecting ducts to absorb more water, the antidiuretic hormone is released, making them more permeable so that more water reabsorption can occur. That is why there is going to be a higher concentration. So let us continue. Here they say, high blood pressure during pregnancy can result in production of urine that contains protein. Describe how a sample of urine could be tested to see if it contains protein. This is easy. We can use the Urea test. This test contains potassium hydroxide and copper sulfate, and it's going to be blue in color due to the copper 2+ in copper sulfate. So the mixture is going to be shaken and it will turn from blue to purple. Then we can know that proteins are present. And here I give you some information about the Urea test. So here we can see protein is not normally found in urine, suggest why high blood pressure could cause protein to be present in urine. Now, guys, remember proteins are big molecules, so they cannot be filtered through at the Bowman's capsule. So if they are filtered, it means there is going to be due to high pressure. So they are forced, the bigger molecules or large molecules are going to be forced through due to the high pressure. So here I say proteins are large molecules, so they are not able to pass through the blood capillaries. This is very important. You state this in the beginning. However, high blood pressure forces proteins to go through the blood capillaries into the glomerular filtrate. This is only possible if there is very high blood pressure. So the proteins are not reabsorbed. Remember, if they are filtered, and they go into the filtrate of the glomerular, they cannot be reabsorbed back, so they're going to remain in the urine. So proteins are not reabsorbed due to their size, so they will be present in the urine. This is an indicator. So basically, when they want to know if they test somebody's urine and they find that it has presence of maybe proteins, they can try to wonder if somebody's kidneys are damaged or if they have high blood pressure. So that could be one reason. So this brings us to the end of question 5. Let's go to question 6. Question 6. The diagram shows one strand of DNA from a section of a gene. So this is the strand they've given us. And they said, what would be the complementary sequence of bases on the messenger RNA produced from this strand of DNA? Remember, this is a DNA strand. We are trying to find a messenger RNA. So the bases we put here should just have uracil and not have thymine. So here I put for adenine. The other side we should have uracil, thymine, adenine, cytosine, guanine, guanine, cytosine, adenine, uracil, guanine, cytosine, and thymine, adenine. So as you can see, this is the one that corresponds to that. Remember, again, this is an RNA molecule, so that is what you should have, uracil and not thymine. So the part B says, the diagram shows a stage during protein synthesis. So they continue to say, which row gives the correct names for the stages of protein synthesis shown and the sequence of bases labeled X. So let's see. Stage of protein synthesis, this is translation. You can see this is the transfer RNA with the codons and the anticodons. You can see there. So this is translation. And then part A labeled X, that is the anticodon present on the transfer RNA. So the correct answer should be here. And I put for you some information here. The codon is going to be on messenger RNA and the anticodon on transfer RNA. So the answer should be C here. Let's continue to the next page. Explain how a mutation can affect the phenotype of an organism. And again, this is a mutation, which is a random change to DNA. So I began off by defining what a mutation is because this is very important. A mutation is a random change to DNA. Now, remember once this DNA has randomly changed, it means the base sequence of the DNA has been changed. So this changes the base sequence of DNA, and this leads to the change in the triplet codon. And again, remember, when translation is occurring, the information in the messenger RNA is going to be read in triplets in order to input the amino acid. So if there is a change to the DNA sequence, the triplet codons are going to be read differently, or they will be in different positions. Therefore, a different amino acid could be brought in the process when translation is going on, and the protein formed could be different. So here I said, this leads to a change in the triplet codon of messenger RNA, and this changes the amino acid sequence in the formed protein. Now, if the protein has a different amino acid sequence, it means it's going to fold differently, forming the 3D structure that could affect how this protein operates. And again, remember, they're asking about the phenotype. So I say this changes to the amino acid sequence in the formed protein, and this will change the structure of the protein. If this protein is acting as an enzyme or acting as a hormone, it could be completely changed that it cannot function, and therefore the phenotype could have a deficiency due to that problem. So let's continue. Here they say, in an accident at a nuclear power station, radioactive material was released into the surrounding areas of land. Scientists investigated the impact of this on a species of butterfly. So you can see this butterfly, really beautiful. So they're going to say, samples of other butterflies were collected from an area near the power station 2 months after the release of the radioactive material. The percentages of butterflies with abnormalities were calculated. The butterflies were then measured in the laboratory, and the percentage of offsprings with abnormalities calculated. So here they're wondering if these abnormalities were basically occurred on the DNA, and therefore the new alleles are going to be passed on to the offsprings. So they continue to say, the experiment was repeated with butterflies collected from the same area 10 months after the release of radioactive material. They say, describe how the scientists could have sampled the population of butterflies in this area. Since these butterflies are in nature, or if they are widely spread, we have to use quadrats to be able to sample them, and these quadrats have to be placed randomly. Then we can count the number of butterflies per quadrat, and then we repeat the process and calculate the mean number of butterflies. So basically, you could throw like 10 different quadrants, add them up together, find the mean. That's the repeating, this part here, repeating. So you have to find the mean. And then we can use the mean in a specific area. Maybe if the quadrant you used was, let's assume, 1 meter squared, 1 meter, 1 meter. So you throw 10 different quadrats, each 1 meter. So 1 meter squared each. And then you count the number of butterflies in each specific area. You add up the total and divide by the 10 in order to find the mean. And after you find the mean, you can use the mean to scale up to the whole area you wanted to sample. So the last part here I said, scaling up to find the total number of butterflies in the whole area. That could be how you do that. So let's continue to the next part. The results of the investigation are shown in the table. Here we have the butterfly generation, adults and the offsprings, and the percentage of butterflies with abnormalities 2 months after the accident and 10 months after the accident. We can see that there are a higher percentage of abnormalities 10 months after the accident for both adults and the offsprings. Also, we can see that the offsprings always have a higher percentage in comparison to the adults, whether 2 months in or 10 months in. So the question here says the scientists concluded that increased level of radioactivity has led to an increased rate of mutation of DNA. Discuss the scientist's conclusion referring to data from the table to support your answer. So from the information we have seen, you can see that there is an increase in abnormality among butterflies collected 10 months due to long-term exposure. So after 10 months for both, there is a higher percentage of abnormalities, and this could be due to long-term exposure. We can also see that the offsprings, so here I say there is a larger increase in abnormalities among offsprings compared to adults 10 months, within 10 months. So there is very high in comparison to that. The percentage change here in comparison to that percentage change, this one is very great in comparison to that. So we can say that this is because, the reason is here, this is because both parents might have mutated genes, and if these genes end up, or these the DNA ends up in the offspring, it means the offspring now receives a higher percentage because it's coming from both parents. So I say this is because the mutated genes are passed on from both parents to the offspring, and this could increase the chance of them having more mutations. Also, parents may have a recessive mutation, or they could be heterozygous. Remember, if there is a heterozygous situation, the recessive allele is not going to be expressed, so you cannot know that. Or, so in this case, this could be passed on. It's not going to be evident in the phenotype of the parent, but it could be evident in the phenotype of the offspring if the offspring ends up being homozygous recessive. So other factors may be leading to further mutations among offsprings. So maybe some other factor they didn't consider could be the reason leading to that. There was no contra-experiment was done, so these conclusions may not be valid because a contra-experiment is required to ensure that any change that is being observed is solely due to the independent variable that is being studied. So here I said radioactivity should be continuously monitored because it can decay. So maybe over time they can observe the decay and see if the effects subside. Also, the total number of butterflies sampled is not included, so the results may not be conclusive. We need to know, because if this experiment is carried out on just 10 butterflies, then that result may not be valid. But if they used maybe thousands or maybe 100,000 or thousands of butterflies, then that could be more conclusive. So we need to know how many butterflies were used or were sampled in order to know if these results are conclusive or not. So this brings us to the end of this question as well as the end of this part. So thank you for being with us. Please do not forget to subscribe. And as I said previously, I will put the link to the part 1 video below the description box of this video. Thank you. Bye-bye.